Hello dear students, this is the continuation of the previous video. Here is the genetics. The genetics which is deals with the heredity and variations. In that, John Gregor Mendel, he considered as a father of genetics. Who considered as a father of genetics? John Gregor Mendel, right? He conducted hybridization experiments for 7 years. See here, for 7 years, work of Mendel. Now, all scientists, for them, it's a base, right? How many days? How many years? 7 years, not a days. 7 years he worked. It's not a simple thing. 7 years he worked on the Pisum sativum, right? 7 years he worked. That's why he has given the number of facts regarding the genetics. That facts are very essential and moreover the right now also like a base for the upcoming scientist, right? He conducted hybridization experiments for 7 years. Here is the 1856 to 1863. This year also have to remember what the starting year 1856 to 1863, right? For span of 7 years he worked on Pisum sativum, right? This is the mid 19th century. Here is the remember this thing mid 19th after only that cell biology and microscopic techniques were developed, right? Here why Mendel succeed, here is the why his work is what you call got success. For that, here is the Mendel work is success, but Mendel is not recognized his work. He has used the math by biology in the first time math biology in the first time. How do you call by mathematical applied statistical analysis and mathematical logics, all the things he had used at the time. It was very new to the other scientists. There is no idea of these all the things. That's the reason, one of the reason Mendel is not recognized. Mendel, his work is not recognized. And high credibility. Here is a high credibility. Utilized a very large sampling size. This is the one of the advantage for the Mendel success, right? Utilized a very large sampling size. That's why you will get the result in accurately. And third one is the precision. Here is the precision is the very important. Selecting 14 true breeding P lines. How many he had taken? 14 true breeding line. Here the true breeding means what? No. Selfing, continuously selfing the plants will get the, when we are cross the two plants, the self pollination only are the one plant. Self, due to the self pollination only you get the true breeding. Right? Representing the seven pairs of contrasting traits. See these three characters, which are three qualities. One is the mathematical approach, high credibility, and precision. Also, these are helpful to the Mendel success in his experiments, right? Who is the John Gregor Mendel? He is the father of genetics, right? Now here is the Mendel had taken the seven characters, right? Seven characters. Here is the first character is the stem height. Stem height. Second is the flower color. And third is the flower position. And fourth is the pod shape and pod color and seed shape, and seed color is the seventh one. These all are the characters. These all are the characters. What are the characters? Character is the expressible feature of an individual, right? Did you see the stem height? We can see, right? That is the stem height is the character. Color is the character. Position is the character. Shape is the character. And pod color also one of the character. And shape of the seed and seed color also one of the character, right? How many? Seven characters. Now what are the trait? Trait means the trait can express the or the define the character, right? It means what? No, distinguishable distinguishable feature of an a character. Tallness and dwarfness. Tall and dwarf. This is the dominant. This is the recessive. Flower color. Violet is the dominant. White is the recessive. Flower position. Axial. See, axial means if it is the flower, that should be as a side. It's the axial region. Terminal means that's a on the top or the particular term end, right? And pod shape, inflated. Inside seeds are there, but this is a inflated. Inflated also called as the full. Full. This pod shape is the full is the dominant, and constructed is the recessive. And pod color green and yellow is the pod yellow color is the recessive. Seed shape is a round and wrinkled is the particular recessive character. And same here, seed color yellow and seed what you call the recessive is the green. See here, difference is where the pod color if you take green is the dominant. You have to use the alleles of the capital G, capital G. For the recessive, should use the small g, should not write the y. Okay. Here is the same thing. Seed color yellow have taken. Yellow. Seed color yellow is the dominant. That's why use the capital Y, capital Y. Here is the small y, small y. And the same here, tall dominant, right? What is the height? Dominant stem height is the trait is the tall. Should use the capital T, capital T. Dwarfness should use the small t, small t. Should not use as a d, small d, small d. Should not use because of the traits we are express are the using as a alphabets. Okay, capital for the dominant and lowercase are the small letters are the dwarfness. Okay, how many characters? 7 characters. How many traits? 14 are the 7 pairs of traits. Here 7 there, here 7 there. One set of is the dominant, one set is the recessive. Dominant is the expressible character. Recessive is the, it won't be expressed, it's a character. I hope you clear. In the next video, we'll continue the next topic.